without further ado, whether you know Stijn from PubMed or from YouTube, please join me in welcoming him to the stage.
Thank you, RL. I'll slowly put this on the stage to really bring up the tension. Hi all, this is very professional. I have my thingy on. It's great to be here. Today I'm going to give you a presentation about endothelial cells. It's, in my opinion, sometimes an overlooked cell type. It's sort of just there. But I hope after this presentation I can transfer some of my appreciation for these cells to you. So let's get into it. These are the endothelial cells we were talking about. And we can see in red some V cadherin and in green vermillance factor. But to me, the main hallmark of an endothelial cell, the #1 marker, is the fact that an endothelial cell is surrounded by other endothelial cells, that it holds on tightly to each other. An endothelial cell on its own, yeah, it's not really, it doesn't do much, but only if it's part of the monolayer that it's part of, it functions. And that monolayer separates our blood and our tissue. And on that location, it has many different functions. In the exchange, in transmigration, or in hemostasis. And to put it in the context of, let's say, blood and sanquin, that means that every protein that passes in and out of the blood goes through an endothelial cell. Any immune cell that needs to get to an area of inflammation or to a tumor, to infiltrate into a tumor, needs to be let in by the endothelial cell. And any therapeutic antibody or personalized viral vector gene therapy you have ever given to someone needs to be actively migrating to these endothelial cells. And in fact, over 90% of those products are broken down by the same cells. So it's quite an important cell with many different functions. So how does it know what to do, when to do what? It's able to do that by responding to a wide variety of signal molecules. Nutrients, growth factors, hormones. And then the endothelial cell accepts these signals, processes them, and makes sure to execute the correct outcome. And it does so by an impressive network of proteins that you see on the screen right now. Actually, the most that we measure in a cell type in our proteomics group. And these proteins right now, sort of 10,000 of them, are in each and every one of our endothelial cells, happily interacting, to make sure that we can all sit here. And this protein network is also the basis of my thesis, of my research. And it's understanding the endothelial response. If you give a certain signal, which proteins are affected. If we give, if there's a disease or a dysfunction, what protein is then the cause in all these 10,000 proteins? How do we start looking for them? That is the main part of my thesis. Now, as Arjen said, I'm very happy to say that everything was published in my thesis. So if you really want to read the technical side, you can look it up. But for today's presentation, I thought, I'll just pick some highlights, some things that have fascinated me, and hopefully also are interesting to you. So I divided this in 3 bite-sized chunks, and we'll start with endothelial inflammation. So I said that an endothelial cell reacts to signals from outside, and one of these signals are cytokines. So what did we do? We take a whole bunch of cytokines and basically threw them on our endothelial cells to see how the proteins change, and that's what you can see here. Some responses are small, some are larger, some are very similar, some are very different. But it becomes interesting when we start looking at the top 2, TNF-alpha and interferon gamma, and if we start combining them. So this is a PCA. It tells us something about how proteomes are different or similar. So this is the control, that's our steady state. If you give interferon gamma to them, they have a specific inflammation state. If you give TNF-alpha, there's a different inflammation state. But if you combine both cytokines together, we get this third inflammation state, a third response, which is the combination of both, but also more, a synergetic response in these endothelial cells. And that's what you're looking at on screen right now. The roadmap, let's say, of synergetically inflamed endothelial cells, and some of the functions, or some of the pathways, processes in that network. Now, I'll go to an example, because that I think makes the point easier. So this is CSL5, it's a chemokine, it's used to attract other immune cells, leukocytes. And we see the RNA and the secreted protein. And as you can see, if you give interferon to it, nothing happens, no expression. If you give TNF-alpha to it, there's an increased expression, but interesting when we combine both cytokines, even though interferon gamma on its own did nothing, together there's this synergetic increase of both cytokines. And what I find really fascinating in this example is that this is C-cell 8. It's a very similar molecule, but the pattern of induction is completely reversed. So in this case, interval HALA does something, but in TNF no. But together they're still synergetic. So it has a very fine-tuned regulation. And the third example I want to show here is IL-6. And I want to show it, it's a cytokine, inflammatory cytokine that we know very well, because the RNA in this case does barely anything. But in the secretome, so what's secreted, we see a large increase. And I think these three highlight very well the intricate regulation of how an endothelial cell signals to its outside. And that we can also see in the membrane receptors. So these are all the membrane receptor proteins that are upregulated after TNF alpha or changing. These are the ones on the interferon gamma, you don't have to know them. This is then after the synergetically introduced one. So it means if you have both cytokines present, there's a massive remodeling of our endothelial membrane, a massive remodeling of the interface with how it signals to its outside. And I think it very nicely shows that endothelial cell is not just a wall or a divisive barrier between blood and tissue, but an active part of it that will change dynamically based on what you give it to it. Now it's great that it's dynamic, it means that we can all sit here, but sometimes it makes things hard to study. That brings us to endothelial heterogeneity. So our endothelial cells are everywhere in our bodies, but they are different in different organs. These are the classical examples I took from somewhere. In the blood-brain barrier, for example, where we want little exchange, very tight junctions. While in the liver and our kidneys, we want lots of exchange of nutrients, waste, reabsorption. So those endothelial cells are very porous. There's lots of fenestrate to facilitate that exchange. But I want to put a mark there. Especially the specific markers that you see on screen here are mostly determined from studies on mice and from transcriptomics. And we have just seen, well, mice are not humans, but also transcriptomics is not necessarily proteomics. So we set out to determine the human proteomes of these different organs. So we got all these endothelial cells from different vendors. It was quite the exercise. And then we did the proteomics on them. So the results came in, and we were very happy to, or very anticipating the great organ-specific, highly specific signature. And then when we run the experiment and it came back, it was all just a bit random. It was just a bit, well, not organ-specific. We did see changes, mostly between small proliferative cells and large differentiating cells, but it was not really organ-specific at all. Now there were some proteins, and I want to highlight some specific markers, some proteins that were different. For example, the SPOC1 missing in the cardiacs, CPE missing in the UVEX, and finally CD36 and the liver. And I want to focus on the CD36. Because CD36 is indeed, if we look in literature, sometimes referred to as liver-specific. That's good, because that's what we find as well. But then also, people also say they found it in the muscle cells, or it's predominantly in the lung, in the heart. And then we also find people that say it's microvascular and others say it's lymphatic. And even in our own study, we only find it in 2 out of 3 donors. So every study finds it specifically, but all studies find it everywhere. So which means, to me, that we simply do not really know yet. We do not know what actually this specificity is. So why is it so difficult, so challenging? Why are there so limited differences? Well, maybe organ specificity is confined to a few proteins. But it's not really in line with our transcriptomic study or the phenotype, that's quite different. But maybe mice transcriptomics is not human proteome. Transcriptomics is more transient. It's more variable. It needs multiple steps to become a protein. And protein, maybe it middles out those differences. But the third and most common, I think, or most logical explanation here is that ECs adapt to the microenvironment. I thought they were dynamic. So the moment you put them in an in vitro culture, with your in vitro setup, they will adapt to the setup and they all become sort of the same. And if you're sitting there thinking now, well, glad I don't work on the endothelial cells because, well, they adapt to things and makes it very complicated, maybe consider that all cells, to a certain extent, will adapt to their microenvironment. Everything is sort of fluid. And I think in vitro is great to study basic molecular biology, protein-protein interactions, pathways. But if you're trying to make the translation to in vivo, well, that should be careful. I think this is what this shows. Are there no situations where you can say something with in vitro about in vivo? Well, let's move on to von Willebrand's disease. For this part, I have to introduce Sophie and Ruben. They work at Erasmus. They're very bright scientists, but maybe even more importantly, very pleasant people to work with. And they like von Willebrand factor and the disease of von Willebrand. So, together with them, we did a project. And it's our on von Willebrand factor. So von Willebrand factor is in these green cigar-like bodies called Weibel-Palade bodies. And von Willebrand factor is a long protein, but in these bodies it's very tightly coiled, packed up like a spring, basically. And the moment there's endothelial cell damage or a hole in the vessel wall, then the surrounding endothelial cells will release these Weibel-Palade bodies. And in this moment, von Willebrand factor strings will unravel alongside the vessel wall. And these long strings, as you can see on screen here in the in vitro situation, are very sticky. So platelets will start sticking to them. And they will start activating other platelets, sticking to other platelets, and form the platelet plug that will plug the hole in the wall. And that's the very first step of hemostasis. And von Willebrand factor is an extremely important protein in this process. Well, how do I know that? Well, von Willebrand disease is the most common bleeding disorder. And it's determined as that the phenotype is insufficient, so too low concentration, or inefficient von Willebrand factor, which means that the protein is faulty. And in a lot of cases, for a lot of patients, we can say, the reason for that is that von Willebrand factor itself, it's a mutation or a variation, it's just not working correctly. But there's also, it's a wide variable disease, and lots of patients here that, well, they're fine, their von Willebrand factor is fine, it's healthy. So, for those patients, we have to broaden our scope beyond von Willebrand factor. For example, to our endothelial cells. Because if you release, for the release of Weibel-Palade bodies, you need a whole spectra, a cocktail of proteins, of which some of them are shown here. And Ruben and Sophie, they came across MEDI. And MEDI activates these RAPs. So the link was made. Those MEDI then influenced the secretion of von Willebrand factor. So what did they do, in that sense? They found patients that were MEDI deficient. And these patients have severe neurological development issues. They do not become old. Von Willebrand disease is not their problem. But we can take those endothelial cells and use them to see if von Willebrand secretion is affected. And to make a very long story very short, our proteomic studies, together with a lot of work by Sophie, most work of Sophie on the functional assays, we were able to show that indeed MEDI is responsible or required for von Willebrand secretion. And that could be interesting then in those patients of which we do not know the reason. Maybe we should look at MEDI at some cases. And I think the lesson that I learned from this, maybe not necessarily on the technical side, but also the project that I did together with people. So I was told this is not going to work, so we'll see. We are all very specialized in our own cell types. We know all the papers. We are very skilled in the techniques we use, and you need to be very specialized in your own niche to be at the forefront. But biology is always interacting on every level, protein to protein, pathway to pathway, cell to cell. Our entire body is a vat of interaction. So in my opinion, to truly understand biology and to truly make those advancements, as we are all extremely specialized, it's ever more important to bridge between those specializations. And the only way you do that is by actively working together with people outside of your niche. And I think that's a nice sentiment to end my presentation on, but not before two things. One, thanking everyone here for that helped me over these past years, especially Arjen and Maarten, of course, and everyone at the MH department, medical priority. And two, so I'm not a man that has very big dreams or like a 5-year plan where my life should be. But I've always had in the back of my mind, it would be cool to be on a stage in a theater, talking about something I like to an audience. And I happened to find myself on a stage in an actual theater with an actual microphone. So of course, I'm going to take that opportunity and do something stupid. So I have no idea how this is going to work. I have no idea if it will sound good, might not at all. But I think sometimes, yeah, you gotta just try. This is a love song. This is a sad song. From the moment we met, I knew that we belonged. Ever since I saw you in that red laser light, I knew I had to be with you. You'll be my delight. I know you in and out, know how you respond, know your curves, your junctions and edges. Yeah, we share the bond. Endothelial cell, how much I love you. Endothelial cell, now I have to go. Endothelial cell, I hope you'll be happy. Find someone to take care of you, now that I am gone. No one seems to care for you. You are just there. Even though you're there, even though you influence all these processes, almost anywhere. Inflammation, hemostasis, kinases and phosphatases. Look at those and stats and gaps. You have so much, but they don't have. You are no T-cell, no macrophage. But don't you ever forget, if you be gone, we all be dead. Endothelial cells, how much I love you. Endothelial cells, now I have to go. Endothelial cell, I hope you'll be happy. Find someone to take care of you, now that I am gone. You and I are not so different. I often feel like no one cares. I just do my little thing. No impact on anyone, anywhere. And the world seems so harsh, so angry and self-absorbed. We seem to be lacking some human affection, losing human connection. But then I'm reminded, an endothelial cell is only an endothelial cell when by other endothelial cells it is surrounded. An endothelial cell can only thrive if hold on tightly to each other. So let that be an example on how we should treat one another. The endothelial cell, how much I love you. The endothelial cell, now I have to go. The endothelial cell, I hope you all are happy. I hope you know that you are worthy, that you matter and that you belong. And thank you for listening to my presentation.